Sanju Nanda presenting you another important and useful episode on a topic in formulation development which will lead to a stable and good quality finished cosmetic product, namely preservation of cosmetics. Preservatives and microbial control of cosmetics make an inseparable part of cosmetic technology paper, which is generally taught as a part of 5th or 6th semester of B.Pharm course throughout India. The contents of this paper shall be deliberated in two parts, owing to the importance of this topic. The first part shall focus on discussing the sources of contamination in cosmetics, introducing the role of preservation, their types, and their evaluation. Let's start this episode while taking a look at what we are going to learn today. We are going to be introduced to the sources and reasons responsible for making the cosmetic products stale or lose their texture, smell, or color, about different chemical classes which can help in keeping the microbes at a bay, and the various efficacy tests given under different regulations. Module 1. Introduction. Everything that has been manufactured in this world is susceptible to decay, and cosmetic products are no exception to this doctrine. Cosmetic products are more important in this context as they are used by a lot of consumers. Actually, they exist in the same form as any topical medicated formulation exists, and by and large, similar excipients are used for making the basic formulation. Products formulated by cosmetic chemists are intended to perform a variety of beautifying functions such as accentuating facial features of the lips, eyes, cheeks, reshaping hair, delivering fragrance, softening the skin, imparting color to the face and skin, and cleansing the different parts of the body like teeth, nails, legs, and face, etc. Chemists are able to deliver many of these functions by using a variety of chemicals and technologies. Assessing the product's stability is a critical part of this formulation process. Figure 1 shows cosmetic products which are available in different physical forms. To bring out the desired product performance, a formulation chemist uses an optimal blend of ingredients. These ingredients may be from natural and or synthetic sources. Water is the main part of the formulation. The product is formulated and manufactured at an industrial scale and packaged in a suitable container. It therefore is an unsaid expectation that the product shall maintain its integrity throughout its shelf life. Cosmetic products may change in their physical and chemical form over a period of time. Light, oxygen, moisture, etc. may play a destabilizing role in cosmetic shelf life. Since microorganisms are also present in abundance in the ambient climate as well as in raw materials, especially water, they may also degrade the product, resulting in change of appearance, color, and texture of the product. Out of the extrinsic factors, cosmetic products are spoiled most by microorganisms. There are three ways in which the microbiological status of a cosmetic product can change significantly with time. First, microorganisms present in the product at the time of manufacture may reproduce and thus increase the number of viable organisms. Second, if package integrity is compromised during distribution or storage, it is possible that the microbiological status of the product may be adversely affected as a result of the ingress of microorganisms. And thirdly, the product suffers most at the dressing table or bathroom shelf of the user. Dipping wet or dirty fingers in the container by one or more users and lack of awareness about the proper storage conditions spoil the product during shelf life. Cosmetic preparations are largely contaminated by molds, yeasts, and bacteria. with the latter generally favoring a slightly alkaline medium and the others an acid medium. Although few microorganisms can grow below a pH of 3 or above pH 9, most cosmetic preparations are within the favorable pH range and therefore a conducive environment for their propagation. Hence, there are two reasons for controlling the numbers and activity of microorganisms in cosmetic products. The first is to protect the user from any health hazard that may result from the use of microbe containing preparations, while the second is to protect the product itself. Module 2 shall contain of sources of contamination and their prevention. There are several sources of contamination in cosmetics which may vary and occur during processing, filling, and packaging, namely raw materials, water, equipment, environment, personnel, and miscellaneous agents. Microbial contamination may occur during the development or production of a cosmetic or during its use. It may also arise from poor sanitation and improper cleaning of working space and equipments. The raw materials are responsible. These are the most common sources of contamination in cosmetics, and their strict quality control is very essential. In general, all inorganic substances derived from mineral sources such as talc, silica, kisilgohar may be contaminated with microbes, especially pathogenic spore-bearing microorganisms such as Clostridium tetani and other Clostridium species. These materials are essential ingredients of either talcum powders or lotions or suspensions. Plant materials which serve as substrates for microbial growth are gums and carbohydrates such as natural gums like acacia and tragacanth, mucilages, pectins, starches, dextrins, and sugars. They become more susceptible when they are in a wet form. Other materials include alcohols, glycerols, vegetable oils, liquid paraffin, fatty alcohols, mannitol, fatty acids and their esters, including animal and vegetable fats, oils and waxes, steroids like cholesterol and ergosterol, proteins, peptones, amino acids, vitamins, etc. Further, all solvents, whether aqueous or non-aqueous, used for processing should also be screened for microbial contents. Thus, it must be clearly understood that firstly, raw materials of good quality should be used, and secondly, care must be taken to discourage the growth of microbes in raw materials during the period of storage, that is, before manufacturing, especially more so in case of raw materials derived from plants. Water is the most common source of microbial contamination of cosmetic products in a number of ways. Ordinary tap water is loaded with a variety of aerobic and anaerobic microorganisms, and its use should be highly discouraged. Freshly prepared, deionized, or distilled water should always be used. Quality of water is important not only when it is used as a raw material but also when it is used for other purposes, for example, like washing the containers, equipments, workbenches, etc. It should also be remembered that water can support the growth of microorganisms and therefore excess water content of cosmetics is also undesirable. Equipments of any type, like processing equipment, which are liable to contribute to the microbial contamination of a cosmetic product, are more than one ways. Contamination may occur if the equipment surface has not been properly cleaned. This may be especially true with abrading surfaces such as those of triple roller mills or those of mixers and turbines. This problem is more commonly seen with vegetable drug excipients such as gums, resins, mucilages, etc. Also, Water or any other solvent which is being used for cleaning and washing must not be the source of microbial contamination. Whenever heating is involved with natural or proteinaceous drugs, care should be taken to clean the equipments after the operation is over. If it is not done, so a remnant layer of proteins may protect the microbes which are hidden under it from the cleaning or sterilization operations. Storage tanks, pipeworks, joints, filters, ion exchange resin beads, and reverse osmosis equipment for demineralization must all be sanitized regularly. A 0.25% formalin can be used to sterilize the microfilters and resin beads. Care must be taken to flush out the solution thoroughly with sterile water after treatment. The next topic is air or environment. Environment in the form of unfiltered air is the potential source of any type of microbial contamination and can affect a cosmetic product during all stages of production, testing, and packaging, and sometimes is a prominent factor even after packaging of a product in its final container. Therefore, operation should be carried out in a clean environment and the air should be filtered and made free from any suspended particulate matter. Rigorous cleaning of wall and ceiling in humid areas is essential since such areas favor mold growth which spread easily by air currents or moisture dropping into different vessels. Wall and floors should be washed with prescribed cleaning agents and disinfectants and should be dried before commencing any operation. Table 1 shows some common microbial contaminants in cosmetics. The bacterial infection is by Acinetobacter, alkaligenes, Bacillus, diphtheroids, Enterobacter, Enterococcus, E. coli, Klebsiella, Micrococcus, and Proteus. The fungi includes Adsedia, Alternaria, Aspergillus, Dermatium, Nitromyces, Penicillium, Rhizopus, and the yeast include Candida, Mornelia, and Torrelia. Personnel. The good health of workers contributing to the production of cosmetics is equally important, especially for the fact that otherwise the workers might be contributing to the overall microbial contamination of the product by way of their hands, breath, hair, epidermal cells, nasal secretions, saliva, etc. Therefore, not only the workers are checked for various diseases at the time of their appointment, but also be subjected to medical checkups from time to time. They are also expected to wear protective headgears, face masks, hand gloves, etc. Miscellaneous agents include, apart from the sources outlined above, two important sources of microbial contamination are the container of the product and number two, the users of the product. Proper sealing of the product from environmental hazards such as air, moisture, etc. is important and hence the containers or packaging materials in which the cosmetics are packaged should be capable of doing so. At times, the consumer herself or himself may contribute to the microbial contamination of a cosmetic in many ways. Even when all precautions are taken to design and deliver a well preserved cosmetic product to the marketplace, there is little that can be done if a product is abused by the user. Some of the most common abuses are diluting the product with water, transferring products to other containers that may not be suitable, storing the product under hot or very humid conditions, or using another person's cosmetics. Microbial contaminants in cosmetic after use includes organisms such as Citrobacter fruendi, Enterobacter species, Pseudomonas species, Serratia species, gram-negative rods, fermentative as well as non-fermentative, Bacillus species like Staphylococcus epidermidis, Sarcenia species, diphtheroids, yeasts and molds. Hence, the policy should be such that contamination should be avoided at every stage and incorporation of preservatives should be reduced to minimum. It must also be clearly understood that preservatives must not be considered as a substitute for good manufacturing practices. The following genera may be classified as health hazards: Pseudomonas, Proteus, Staphylococcus cereus, Penicillium, Aspergillus, Candida, and Monilia. The following organisms may be classified as objectionable for damaged epithelium: P. originosa, S. marcescens, Klebsiella species, P. multioverns and P. putida, S. aureus, C. perfringens, C. tetani, and C. novel. For cosmetics of the eye, P. originosa is listed as always objectionable, while other Pseudomonas species and S. aureus are classified as usually objectionable. Always objectionable is based on two factors, namely pathogenicity of the organism and site of use, while usually objectionable also includes an additional determinant apart from the above two, that is the state of health of the user. Safety limits. The cosmetic products can be accepted as safe when the microbial counts is limited as mentioned below. A. Products intended for general use. Total viable counts of aerobic bacteria, yeast, molds should be less than 1000 colony forming unit per gram or milliliters. B. Products intended for specific use in the US area and those recommended for use for babies. Total viable count of aerobic bacteria, yeasts, and molds should be less than 100 colony forming units per gram or milliliters. Action of microorganisms on cosmetics. A number of adverse effects due to the growth of microorganisms in cosmetic products are evident. These include discoloration of the product, the formation of odor or gas, changes in the rheological properties of viscous products, cracking of emulsions, etc. The visual evidence of growth of pigmented fungi on the surface of a cream can be most disconcerting to the customer who has paid a heavy price to purchase it and had expected some good results from it. Sometimes decompositions may not reveal itself or many months if conditions are such that growth only takes place after the organisms have adapted themselves to the environment. Usage of such products is more harmful, especially so when used on damaged skin. The third module shall consist of methods of preservation of cosmetics. Traditionally, there are six major means of preservation or microbial control of cosmetics. They are: number one, chemical preservatives. These are the most widely used substances for preservation of cosmetic products. These chemicals constitute a minor component of any formula and are usually included at levels of less than 1%. Examples are benzoic acid, sodium benzoate, methyl hydroxybenzoate, propyl hydroxybenzoate, etc. The second way is manipulation of pH. Normally, the pH for most cosmetics is within the range of 3.5 to 9. Manipulation of pH may aid in the effectiveness of the chemical preservatives as well as exerting a limiting influence on the microbial growth of many strains of microorganisms. This technique is effective but somewhat restricted because of possible eye and dermal irritancy. The third way is limitation of water content. Controlling water activity to a level that does not readily support the growth of microorganisms is commonly used with cosmetics whose primary components are oils, waxes, and other materials that do not contain appreciable amounts of water. This approach alone does not prove satisfactory for those cosmetics which may be stored in humid places or in which there are chances that the user may add some amount of water while using these products which contain hydrophilic substances. Chances of spoilage due to the presence of water are reduced if the water activity in a product is maintained below 0.85. The fourth approach is use of hostile raw materials. Incorporation or presence of certain ingredients in a formulation are helpful in preventing the growth of microorganisms or are self-preserving. For instance, colognes and other perfumes fall under this category. Since they contain high percentage of alcohol which acts as a self-preserving agent too. Other hostile materials such as propylene glycol and 1,3-dibutylene glycol can also act in a similar manner when used at fairly high concentrations of 25 to 50%. Certain surfactants may also show antimicrobial activity when incorporated into the final formula. The fifth approach is of aseptic manufacturing. Introduction of microorganisms, whether through improperly controlled process, water, or by unclean equipments, may injure the preservative system. In many instances, resistant microorganisms develop in improperly cleaned and sanitized equipment and grow in products that are considered well preserved. Therefore, all compounding and filling operations must be closely controlled. A proper check on the raw materials and critical packaging components must be strictly exercised. As per good manufacturing practices, it is a recommended practice to have approved cleaning and sanitization procedures for all equipments and to microbiologically monitor these procedures at critical stages of manufacture to confirm that the system is operating as intended. Although not actually a means of preservation itself, aseptic manufacturing is necessary to ensure that preservation is not compromised by the manufacturer. Package design also has a role to play. Ideally, the design of package should be such that it does not allow any interaction between the product and the environment. However, such a package does not exist except in case of single application units. Such packages are otherwise not widely used in cosmetics. Conventionally, jars or wide mouthed bottles are used for creams, collapsible tubes are used for toothpaste and shaving creams, bottles with narrow mouth are used for lotions, packages or collapsible tubes that allow extrusion of the product in one direction, and those containers that employ dispenser pumps are preferred over open jars or tins that are repeated contact or mixing by the user. But it is not possible to pack all formulation in the same manner. Such products must be preserved by use of an appropriate preservative system. Certain essential oils, fragrances are known to possess antimicrobial activity. At the concentrations levels found in many cosmetics, they may or may not be effective adjuncts to a preservative system.